Yes, so thank you very much for having me and for all the interesting talks that we've heard so far. So I'm just going to talk briefly about the future of current flow modeling for electrical stimulation. So next slide, please. The appeal of electric stimulation has always been its therapeutic potential. However, this has been marred by variable outcomes. So for every significant effect of stimulation, there's a null effect. However, thanks to current flow modeling, we can now, as we can see with the previous talks that we've gone through, we can move away from this one-size-fits-all approach where we apply the same stimulation protocol to each individual. And in particular, using models, we can now consider, for example, these three components. So intensity, where are the peak intensities in the brain? Focality, how distributed are these peaks depending on electrode montage? And also direction, what orientation is current flowing in our region of interest? So I'm just going to talk briefly about research aimed at quantifying and reducing the variability in intensity and direction. So next slide, please. So looking firstly at intensity, conventionally what we do is we apply a fixed dose of stimulation, such as 1 milliamp, across all individuals. But we know due to anatomical differences, including things like skull thickness, the intensity observed in the brain likely varies across individuals. Here, what we wanted to do is quantify this variability when you apply a fixed dose of 1 milliamp to the left primary motor cortex, or M1. And what we observed, which you can see in figure 2, is that there was over 100% variability in E-field intensity in that target region, so anywhere between 0.1 volts per meter to 0.3. We can also see that there was substantial variability in the distribution of electric field across our subjects. And finally, which Sven touched upon earlier, which you can appreciate in figure 4, this variability is actually exacerbated by small sample sizes. So for example, in simulated data of 15 subjects, which is still a common sample size that we see in electric stimulation literature, the red distribution, we can see much lower intensities in M1 compared to in the sample in the purple distribution. So you might even speculate that just these two populations may have very different responses to stimulation. So we wanted to explore how, instead of applying a fixed dose, we could fix the intensity at the cortex. And we could do this simply by increasing or decreasing the stimulator output for each subject. What we can see in figure 6 and 7 is that by individualizing dose, we effectively eliminate that variability in M1, and we can also produce more consistent distribution of electric field across individuals. Instead, we've effectively shifted this variability to the stimulator output. Next slide, please. So in addition to intensity, we also wanted to look at variability in direction of current in target M1. So current direction, we typically manipulate by placing either the anode or the cathode over the target site. So when we place the anode over the target, we are expecting to maximize radial inward current in our region of interest, which we commonly assume will increase excitability. On the other hand, if we place the cathode over the target, we theoretically maximize radial outward current, resulting in reduced excitability. So here we looked at applying a conventional montage for targeting M1, where you place the anode over the target and the cathode over the contralateral supraorbital ridge. And we looked at two regions within M1 in particular, the gyral crown of M1 and the sulcal bank. And what we observed, as we sort of anticipated, is that there was substantial variability in the direction of current across subjects, which you can see in figure 2. And this really emphasizes that need for individualized electrode montages. And also notably in figure 3, we can see that when you apply a conventional montage, we appear to indeed maximize radial inward current in the crown of M1. We also wanted to quantify the variance in a different montage known as the posterior anterior, or PA montage. So this montage, instead of placing a target electrode over the region of interest, we actually place electrodes either side of the target. This is believed to result in more consistent current within the target region. However, we can see, looking at figure 5, that when you look across subjects, we have similar inter-individual variability in current direction, both in the bank and crown of M1. However, what we did notice is when you apply this PA montage, we instead have radial inward current maximized in the bank of M1 as opposed to the crown, which we saw with the conventional montage. So this suggests that these different electrode montages may actually deliver radial inward current to distinct subregions of M1, which could potentially limit their comparability. Next slide, please. Okay, so just to bring it all together, what these two studies are touching upon is ways in which current flow models can facilitate that move towards individualized stimulation. And we saw a lot of other nice examples already today. We can individualize dose to fix intensity at the target rather than simply maximize. And we can also individualize montage in order to maximize the radial current in our target. But these results also demonstrate that the optimization problem is multivariate in nature. So it requires a careful balance between several components, including those focality, intensity, and direction measures. And finally, I just want to, little food for thought, which always is on my mind, is the key challenges I think remain for the optimization. And that is, do we actually improve outcomes in a meaningful way if we control for these parameters? Do we see substantial improvements in TES effects as we hope? And finally, one of the significant appeals initially of electrical stimulation is that it was very simple and easy to use, even a take-home method for patients. So with the introduction of things such as current flow models and the required data analyses associated, how do we ensure that this remains a user-friendly technique? Thank you very much.